Welcome to On the Issues. I'm your host, Nicholas Balsi, and today I'm delighted to be joined with Nicholas Wade. He's a journalist and an author, and we're here to talk about a variety of issues in the area of science and technology. I want to thank you for taking some time to sit here and talk to us before your lecture.
It's my pleasure.
I guess the first question would be, as somebody who's well-versed in the area of science, in your books and your columns, what are the recent issues in the area of science affecting the country?
Well, I suppose on the biological side, the one you hear most about is stem cell research. So a lot of new money is going to stem cells from states, which usually have been very reluctant to fund scientific research. And there are a lot of hopes for this technology because the idea is that the best way to repair the body must be with its own methods, with its own cells and signaling systems and so forth, if only we understood how it worked. And it's clear that stem cells hold part of the key for how evolution builds our tissues. But whether those hopes will be realized in the near term, I'm not so sure. I think we have a lot more to learn before we can manipulate the cells to make them do what we'd like them to do.
Now, I guess on the journalism end of addressing an issue like stem cell research in a column, when you're writing, a lot of times with columns and the general consensus in journalism is to be balanced. How do you deal with an issue like that when writing these columns, especially on an issue with stem cell research?
Well, you know, I used to be an editorial writer where you have to just come up with opinions. And I often used to envy the people who are writing the news reports because their job seems so easy to me. Mm-hmm. All they have to do is ask all the relevant players, the people on each side, and leave it to the reader to decide, whereas the editorial writer has to make his own conclusion and decide, you know, who's doing the right thing and who's doing the wrong thing. So for a reporter, the main thing you need to be sure of is that you understand an issue well enough to know who the major players are and often who the minor players are as well, whose voice needs to be heard, and just make sure at least you've checked in with all of them and, if possible, presented everyone's views.
So that would also relate to how the whole religious, moral issues come into play with issues like stem cell research when writing on a topic like that?
Right. So we always try and present both the scientists' view of stem cell research and those of interested ethicists and religious parties.
So when an issue comes into the news or an issue that you want to address in a column, sort of what is the process that you go through to come up with the column as a whole, contacting the people doing a study or things like that?
Yes, if there's some new research, you'll start off with the research paper, and you'll have the scientists who've written it. So that's one obvious source to go to, and then you maybe will have their critics. You may know that some people don't agree with their theory, so you'll check in with the critics. If it's a political story, you may check in with the congressmen, who always like to get their names in the paper. So you should be very willing to tell you their point of view, and if it's a religious story, you'll check in with the spokesman for the Catholic bishops or the Protestant churches or the rabbis, whoever is involved.
Do you find it more difficult to report on an issue that's also political or a strictly science issue?
Well, I think the political issues are probably, in general, easier because they're more accessible. Mm-hmm. So everyone understands a sort of a political fight, and they will read stories that are sort of couched in the form of a debate or an argument. Whereas the science is often a lot more groundwork you need to do because the vocabulary is unfamiliar. Even the most scientific concepts are generally quite sort of easy to follow. Mm-hmm. They're nonetheless clothed in this sort of arcane vocabulary which has to be explained. So there's definitely a barrier there you have to get over.
Now, I was looking up on some of your columns, and I guess moving on to some specific things that you've written about that may be interesting to expand on. One of them being that a gene that governs pain perception was found recently. Could you possibly offer some sort of insight into this new finding, this new study?
I'm just thinking back on the story. That was found in a little Pakistani boy, is that right?
The 10-year-old Pakistani boy. Sort of what this kind of means to science in general, and why it was worthy to actually write a whole column on.
Right. Well, I think it came as a surprise to people who work in pain, because they thought that pain is so sort of deeply embedded in the human body that there will be many genes that set up the system for us sensing and perceiving pain. And it came as, and of course that makes much more complex the task of trying to address pain and find drugs that reduce it, because you've got so many different genes that might go wrong. So this paper was electrifying, because it said, well, it's just one single gene that controls the perception of pain. So that's a sort of deep and surprising insight into how our bodies are constructed, and it also gives you hope that, well, if you understand just this one gene, you can find a new class of painkiller drugs. So that's why that story seemed fun.
Now, use that as an example and take that into consideration when talking about journalism as a whole, because when you write a column like that, you said it has an effect on other issues, other genes, other issues in science. How do you sort of take that into consideration when you're reporting on an issue like that? Do you have to go to all the different studies and progress that has been being made in the other areas that you're touching on as well?
Well, ideally you would do all that, but you're limited by both time and space. So although one would often like to write a long article in which you did touch base with everyone you can think of, and that would be more space than the papers like to give you unless it's a very important story, and also be longer than most readers would sit through. So for most of these, for a large number of sort of breaking scientific discoveries, we'll just do fairly short stories. Then one could always go back to the issue later with a longer feature story if it seems warranted. So if there's any advance with this pain gene, or someone comes at it from another angle, then I'll probably do a feature story sometime.
Okay. Another specific issue to touch on too, one of your recent columns was the possibility of a Jewish tie to Thomas Jefferson.
Right.
How did this come about, and how did you sort of get involved with the issue to write about it?
Well, I guess it started because of the rumor that has been flying around since about 1820 or longer that Jefferson had, I think we read in 1720. I'm sorry, had a secret family with his slave mistress Sally Hemings, and that was published in a local newspaper at the time. But the Jefferson denied it, and a whole string of historians who've looked at it decided it was just a vile rumor put around by the press and was not true, because it was so untypical of Jefferson in their view. So the other matter stood until DNA testing became available, and a historian who's associated with the Jefferson family persuaded several Jefferson descendants to donate their tissues, so that this rumor could be laid to rest. I think that was his expectation. So Jefferson himself had no male descendants, but his paternal uncle, Field Jefferson, had lots of descendants, and their Y chromosome was presumably identical with Jefferson's, because it's passed down from father to son. So their Y chromosome was tested and compared with a descendant of Eston Hemings, who was one of Sally Hemings' sons, and it turned out to be identical. So that more or less established, it made it very likely, though it didn't absolutely prove, that Jefferson did have a family with Sally Hemings. But if you take the genetic evidence and you combine it with the available historical evidence, for example, showing that Jefferson, though he traveled a great deal, was present at Monticello at the time of conception of all of Sally's children. If you combine those two bits of evidence, it's a very strong case. So that story broke, I think, about 9 years ago, and it suddenly came back into the news because a geneticist in England, who'd been part of the original team that analyzed the chromosomes, thought it would be interesting to see where in England Jefferson's family came from. And he was very surprised that at first he couldn't find any of these chromosomes. It turns out to be a very rare chromosome. He could only find it in the Middle East. So that made him think, well, it's absurd to think Jefferson's family came from the Middle East, and the Jefferson family tradition is they came from Wales. So he said, well, I'm going to look harder for this chromosome in England. So there's a special population of people in England who are most likely to carry this chromosome. It's people named Jefferson. So he looked at all the Jeffersons he could get from the phone books, and he found just 2 in the 85 people he tested had this chromosome. So that does establish that that certainly the chromosome was present in England, and that's surely where Jefferson's ancestors got it. But then the question arose, well, how did it get from the Near East, where it seems to be most common, to England? So this could have been 12, 15,000 years ago. It might have been just people migrating as everyone else did. Mm-hmm. Or it's possible that it was in the Jewish community when that was found about 3,000 years ago, and brought either by Ashkenazis or Sephardis to Europe and then to England. But that's in no way proved. It's a mere possibility. And some of the people I talked to thought it was just as likely that it was sort of direct migration from the Middle East. It hadn't necessarily passed through the Jewish community.
So does this issue have a reflection on any others in the area of gene research and science? Could, I guess, maybe the time that this is being discovered, how long, that these things can never be done that quickly, that anything could happen out of nowhere?
Well, yes, I mean, it shows the extraordinary reach of this genetic technology. Mm-hmm. So basically, you can tell who slept with whom throughout history. If you sequence everyone's Y chromosomes and everyone's mitochondrial DNA, mm-hmm. you could put everyone on a single family tree that would go back to the ancestral human population that lived about 50,000 years ago.
I also know that some of your past research has been done on how we can trace our ancestors back. How could somebody like an average everyday person, if they want to look back very, very, very far into the past, how could just someone do that?
Well, there are lots of these DNA testing companies nowadays, and you write to them, and they send you a little kit, which is basically a little swab, which you scrape inside your cheek, and lots of cells come off on the swab, and you put it in a little plastic bag and send it back to them. And they will test it, either they'll look at your Y chromosome or your mitochondrial DNA. That's the DNA in the mitochondria, these little organelles inside a cell. And those 2 pieces of DNA are very important because both of them escape the shuffling of DNA between generations. So they sort of come down unchanged from the ancestral human population, accepting only that they sort of pick up mutations along the way. And the mutations they pick up are very diagnostic of different regions of the world. Because if you imagine people coming out of Africa and very slowly going from one country of the world to the other, at each sort of way along the journey, a new mutation will hit the chromosome. And you can use these mutations to put everyone on a Y chromosome family tree. And that tells you, which lineage you belong to. So basically, the information you get back from these testing kits is that, well, your Y chromosome is sort of lineage L3, or your mitochondria is lineage K. And they'll also tell you sort of roughly where in the world that Y chromosome or mitochondria is mostly located. So that mm-hmm. isn't terribly informative right now, unless you happen to know lots of other people who's gone to the same testing service and know they share your Y chromosome lineage. But even so what? I mean, Y chromosome doesn't do very much except determine maleness and fertility. It's not a particularly interesting chromosome. So I'm not quite sure what people really do with this information. It's sort of nice to have. It's going to mean a lot more in the future, but right now it just sort of tells you roughly where in the world your ancestors may have come from a long time ago.
Yeah, I guess it may be nice to know the background of your actual family and the history, so you know where your family's been, in other words.
Yes, especially when we've done a lot more work, we'll be able to identify many of these lineages much more precisely. So one of the major lineages that's been discovered is one that belongs to Genghis Khan, who, when he founded the Mongol Empire in the 13th century, he was very much into harems. So there were a lot of women with whom he had children. And Genghis Khan's chromosome is found all over Central Asia. And in fact, there are 16 million men in the world today who carry Genghis Khan's Y chromosome. Wow. So that's the kind of sort of specific bit of history that's quite interesting if you happen to find you're a descendant of Genghis Khan. But there are very few chromosomes. There are 2 other chromosomes for which that history has been established. But we need to establish a lot more before you can tie yourself into any recent historical figure.
Makes sense. And now possibly moving on to your actual opinions on some issues as someone who's done quite a few editorials. How about stem cell research? Since you did say it was one of the most important issues affecting the country right now, you have a personal opinion on the issue?
Well, I should be a little careful here, Nick, because since I'm now a reporter, not an editorial writer, the reporters are meant to be as objective as they can, so they probably shouldn't talk about their personal opinions. But I can certainly sort of discuss any issue in a sort of factual sense. Is there any particular thing you want me to?
Oh, well, basically, there's a lot of political issues surrounding it as far as giving funding for it and different things like that. What could stem cell research potentially do for our knowledge of diseases and different things right now in science and health?
Well, what it could potentially do is, and it could make use of the fact that all the billions of cells in our body come originally from a single cell, and all the cells in our body contain all the information from the original egg from which we came. So, and that information is enough not only to sort of design out to create our body in the embryo, but it also has all the information for maintaining it. So if only we could somehow tap that information for when things go wrong, because all the information is basically there to correct it, that would be a much more profound kind of medicine than the things we have at present, which I'm sure in sort of 50, 100 years will be regarded as immensely crude, like we regard sort of burning holes in someone's back as a totally antiquated kind of medicine. So we use the knife, which is very crude, or we use these powerful drugs, which are basically poisons, and these are the best things we have. But they're so unsubtle compared to what the body itself could do. So the whole idea of what is called regenerative medicine is to use the body's own systems. And a very central piece of that is stem cells, because the way our tissues are constructed, it seems, is that you have a guardian stem cell for each kind of tissue. And this stem cell keeps on throwing off new cells which turn into the adult cells of that kind of tissue. So supposing, I don't know, you have liver damage because you've drunk too much and the liver cells have been killed. Well, wouldn't it be great if somehow you could find a liver cell which would generate more natural liver cells, and that would repair someone suffering from cirrhosis, which is basically ultimately a fatal disease. Right. That's what we hope we can do. So far, I would say we haven't made a great deal of progress.
That's my next question was going to be the future of this. Do you see it being supported anytime in the near future?
Well, the research is definitely very well supported, but one question that some scientists have is whether we aren't going about it the wrong way, because nature does regenerate tissues, not in us. Well, if you get the tip of your finger cut off and you're a child of less than 12 years, your finger will grow back. That's about our best kind of generation. But there are other animals that can regrow. Well, a lizard, if you catch its tail and it breaks off, a lizard will regrow a whole new tail. And deers can regrow their antlers. And there's a creature called the axolotl that regrows legs, regrows its heart, regrows most of its body. So why have we lost this power of generation? It's probably something to do with cancer, because we live so long that evolution has chosen to give us very strict controls on cell growth to prevent cancer. And the trade-off is that, well, we can't regenerate so well. But the way nature does regeneration in these cells is that it does not start with stem cells. It starts with the mature cells, and it walks them back to being stem cells, and then it grows the tissue again from there. So if evolution in its wisdom has said the way you do this regeneration trick is you start with the mature cells and you walk them back to being stem cells, it seems to me awfully unlikely that we're going to be successful doing it the opposite way around, i.e. starting from stem cells and going to mature cells. And the reason we've got into what may be a sort of intellectual trap is the paradigm of bone marrow transplants. So the one place where stem cells definitely do work is if you have leukemia, say. You can have all the cancerous cells cleared out of your bone marrow, and the stem cells harvested from a donor, the stem cells that make blood, can be injected back into your bloodstream. They home in on the bone marrow, on your bone marrow, and recreate your blood system for you. Mm-hmm. But blood is a liquid tissue, and it may be that this is the one area that stem cells do work. And for repairing solid tissues, the cells need to know where they are, exactly where they are, and what they're meant to be doing. And that information may be programmed into them during embryogenesis. So when the fetus is formed, the cells that make the liver, as they turn into the liver, they have programmed into them the idea, well, you're in the upper left-hand quadrant of the liver. Don't stray away from there. That's where you're meant to be. If you leave there, please die, because we don't want you around. That positional information is not in the stem cell, and it's very hard to recreate.
Oh.
So if we're going about it the wrong way, then we're not going to see many results.
So you see the problem more in the way that the research is being conducted than necessarily support for the actual research currently?
Well, first I should say this is a minority view held by just a few scientists. And that most scientists are very high on stem cells. They believe results will come sooner or later. They point out correctly that all research is difficult. It always takes a long time because you're right at the frontier of knowledge. So it's simply too early. It's too early to say, from which avenue success will come. And the obvious strategy is to try everything: to try adult stem cells, try embryonic stem cells, to look into this regeneration, to sort of do it across the board, because basic research is unpredictable. You can never tell what will be successful.
Now, last but not least, for the future, if the government, as far as the actual administration, doesn't necessarily support stem cell research and its concept, will the research still continue, or does it need that government support to proceed?
That's a complicated question. I mean, the government, Bush administration, does support research with adult stem cells, and it also allowed embryonic research with embryonic stem cells to begin. Its actions, I think, are sometimes misportrayed. What it did do was put a restriction on the kinds of embryonic stem cells you could use. They had to be ones that had been created before August 9 of 2001, as I recall. So that gives scientists enough embryonic stem cells to get started, to do the sort of most of the basic research they need. Eventually they will need to create other stem cell lines. It would be very helpful if they could do that with government money, which at present they cannot. You cannot, with government money, create new stem cell lines. So that is already a hindrance and will become more and more of a hindrance. However, it hasn't stopped the research completely, because in reaction to the government's policy, lots of private money has has come into stem cell research. And California in particular has voted $3 billion by referendum for research primarily on embryonic stem cells. So it definitely will be going ahead, and it may even be that the stem cell scientists, because of people's disagreement with the administration's policy, will be able to raise more money than they would otherwise have done.
All right. Well, I want to thank you for taking time to
You're
come
very
on the
welcome.
show and discuss these topics.
It's my pleasure.
Thank you.
Thank you for those questions.
And make sure you tune in to On the Issues next week. And again, I want to thank Mr. Wade for coming on the show. And tune in next week. Thanks.